Now it is time for us to move into the announcement of our new grants. We are very pleased to be funding just over $900,000 to two very exciting research projects this year. And this is bringing us that much closer to our 30th anniversary goal of committing to $20 million in research funding. So first up, we are very pleased to announce funding for Dr. Francesco Muntoni's research into AAV gene therapy for DMD-related brain dystrophin deficiency. Dr. Muntoni is a professor of pediatric neurology and the director of the Dubowitz Neuromuscular Centre at the UCL Great Ormond Street Institute of Child Health and Great Ormond Street Hospital for Children in London. Dr. Muntoni has also been director of the Genetic Therapy Accelerator Center since 2022, which is a new gene therapy translational research partnership based at Queen Square Institute of Neurology and in close collaboration with the UCL Great Ormond Street Institute of Child Health. He's also chief investigator of the European Brain Involvement in Dystrophinopathy Consortium. Tonight, we are pleased to welcome Dr. Muntoni's colleague, Dr. Konstantina Teterou, a research fellow at Dubowitz Neuromuscular Centre of Great Ormond Street Institute of Child Health. Dr. Teterou holds a PhD in neuroscience, focusing on molecular and inflammatory pathways and their role in potential new therapeutic approaches. Her main research area is the brain development in Duchenne muscular dystrophy. Dr. Teterou will present details of the research grant, AAV gene therapy for DMD-related brain dystrophin deficiency. Thank you so much for joining us tonight. We really appreciate it, and I'll hand it over to you.
Thank you very much for your introduction and the invitation, and thank you very much for supporting our research this year. And congratulations on your 30th anniversary. I will be sharing my screen.
Thank you so much. Are you able to share? There you go. Wonderful.
Yeah. So Professor Muntoni was not able to join online today, but he recorded a short video that then I will present the aims of the research project.
Hi, my name is Francesco Muntoni. I'm a pediatric neurologist from London, and this video just to thank Duchenne Canada for the grant we've been given on AAV gene therapy for DMD-related brain dystrophin deficiency. I work in London, in the UK, and unfortunately, at the time you see this video, I am flying and unable to take part in person to this presentation. But I'm going to briefly present the team that will address the issue of our grant. And as you are aware, DMD is not only a muscle disease. There are some brain comorbidities, both cognitive involvement and neuropsychiatric manifestation. Luckily, these problems are not present in every individual with Duchenne or Becker, but when they are present, they cause difficulties. And we want to better understand how to deal with them. Which question will our project address? Firstly, how much the behavioral issues detected in the mice relate to developmental role of dystrophin in the brain. This is important. How much it relates to when the dystrophin is present during fetal or early development, or how much can be rescued postnatally. Also, which are the protein complexes secondarily affected when dystrophin is lost in the brain? We know very well what are the protein involved with dystrophin in muscle. We know very little regarding what are the proteins dystrophin interacts with in the brain. And finally, can AAV gene therapy not only improve muscle function, but also the brain function in mice with Duchenne muscular dystrophy? The team that will be addressing this is a team here in London at the Institute of Child Health and Institute of Neurology. In addition to myself, there is Professor Jo Ng. She has a lot of expertise on AAV gene therapy. The Professor Simon Becks is a world expert regarding deep phenotyping from a behavioral perspective on animal models, and Konstantina Teterou, who will be now explaining to you more in detail how the work will be done, is a talented researcher who has been working in Duchenne brain now for a few years as part of my work. I'm sorry I cannot be there. I wish you all the best, and thank you again for the generous support for our work.
So dystrophin, there are different isoforms that they are expressed in the brain, and that are highlighted in blue. And these are DP427CP, DP140, DP71, and DP40. These are different isoforms that they are not expressed in the muscle. Dystrophin in the muscle has different protein complexes that they are presented on the left-hand side image. And they are different from the complexes that they are described so far in the brain. The protein complexes in the muscle, they have been well characterized, although the protein complexes in the brain, they have not been characterized fully. In our study, we're going to use a humanized MDX52 mouse model, which is a new mouse model with humanized region on exon 49 to 53 of the DMD gene. The MDX52 mouse model has been well characterized on the behavioral phenotypes this mouse model presents, and two of the main features, behavioral features, are the fear conditioning and robust anxiety-related responses on this mouse model, as well as emotional disturbances. Firstly, we're going to investigate the developmental role of dystrophin, and in order to do that, we're going to induce deletion of dystrophin with tamoxifen at embryonic stage, neonatal stage, and in adult stage. And then, subsequently, we are going to evaluate the neurobehavioral consequences by inducing the isoform loss at 3 developmental stages. Moving to the second aim, on the dystrophin protein complexes and interactors in the brain. This picture depicts what the current knowledge of dystrophin interactors in the brain. Within the BIND consortium, the Brain Involvement in Dystrophinopathies European consortium, we were able to identify a larger number of proteins that they interact with the 3 main isoforms that they are expressed in the brain. The significance of this research was that most of these proteins, they were related to the neurobehavioral comorbidities that the patients develop and present, such as cognitive impairment, autism, psychological disorder, and learning disorders. So the second aim is to evaluate the dystrophin interactome differences with the DP427 and DP140 isoform loss prenatally and neonatally in the humanized new mouse model. The last aim of the project is a brain-targeted AAV gene therapy, where we're going to inject different AAV gene therapy constructs at the neonatal stage. And then when the mice will reach adult stage, between 8 and 12 weeks, we're going to perform a battery of neurobehavioral tests to test cognitive and emotional difficulties. And then we are going to extract the brain, and we're going to test protein expression and RNA transcript levels on the mouse brains. Therefore, the aim of this is to evaluate the brain dystrophin expression to ameliorate neurobehavioral deficits in a dystrophic humanized mouse model. I would like to thank you all for your attention, and this is a team from UCL and also the BIND consortium colleagues for the neurobehavioral results that they have been achieved. Thank you very much.
Thank you so much for the presentation.